est ravi d'avoir Rohwer ici. C'est grâce à Christelle qui avait fait son postdoc chez Forest. Forest, c'est celui qui a la première fois décrit le virome par métagénomique, les virus par métagénomique en séquençant tout. Il vient de l'environnement et il a appliqué maintenant son travail sur les prélèvements humains et a fait des découvertes très spectaculaires, en particulier sur les virus de plantes qui ont inspiré beaucoup de travaux ici, et puis sur les phages. And we recently used your data on phage to show the resistance within the phage that you find in cystic fibrosis. It's a work by Jean-Marc. So we are looking, when you publish, we are surveying you to see what you're going to do from here. So we are very happy to get you here.
Okay. Bonjour. That's all French. That's I'm done. We're going. If you have a question, just stop me because I know it's going to be a little harder versus your French. All right. So what I'm going to do today is I'm going to actually just go through where we are in the Human Virome research and kind of what we're looking forward to doing in the Virome research. And I'm more than willing, I know you guys have seen some of this, so ask me questions about things that you might think about. And these are just some terms, just when I say phage, I explicitly mean phage, so things are affecting bacteria. The rest of the time I'll say viruses. We talk about VLPs a lot in this field, and microbes, I don't differentiate between bacteria and archaea. And there's going to be a fair amount of lytic and lysogeny behavior, as well as the difference between proviruses and induction. All right, so my starting idea always is that the viruses are the coolest thing out there, much more exciting than the bacteria or the humans. Okay. And there's something like about 10 to the 31 of them on the planet, which makes them numerically the winners. We know that there's at least 100 million species, whatever you want to call a viral species. That's an underestimation. And much of my work has been really in this area, which is right here, understanding the dynamics of viruses with different hosts and how they drive this population where you get, one strain will come abundant and then it'll be killed off by a virus and that will lead. And that seems to work on bacteria, archaea, and human cells also. Okay. The other thing is that what we're really coming to is this idea right now that essentially most of the genetic diversity on the planet is viral. And by most, I mean about 90%. So it seems to be the major reservoir of genetic diversity out there. And of course, we're interested in from diseases. Now, this is what they look like if you're not used to this. This is really our starting point all the time, is that here are your bacteria and then all the little particles are going to be the viruses. And what we figured out in environmental systems is that they really drive these sorts of top-down, kill-the-winner dynamics, right? And you may have heard this proposed evolutionarily as this thing called Red Queen. So the idea being that you have to get resistance to your viruses to survive, and that there's this arms race going the whole time. So the starting point is, what about in humans? And this has been going on in the lab forever, but it's only recently that people actually decided that the microbiome of humans were interesting, which means that now you can get money to study it. And so the first study was with monozygotic twins. The main lab that we work with is Jeff Gordon here. If you know his work, he's the one that's been doing, he did the first obesity, whether the microbiome was different in obese people versus not. And what they have is this really, really nice data set of where they've been following twins for literally decades in Missouri. And this was a psychology test, actually, to see how they behaved, how they thought and everything. And since then, they've actually tapped into this and started doing the biology of these twins and figuring out. So this first set is monozygotic, which means they have the same DNA, each twin does, and their mother, and then we looked over a year period and just did this normal thing of isolating the viruses and sequencing them. background from the microbial point of view is that co-twins and their mother were very much like each other when it came to the bacterial community. So that there was something about living in a family, either the food you were eating or exchange of bacteria, that really made you very similar to your mother. All right, now when you look at the viruses, what you get is that the same thing that we normally see is that you sequence the viruses, check them against your favorite database, and then ask, what do I have there? And what we always find is that there's basically no hits to the viriome, so essentially everything is unknown, the virus part of it. And then the microbial part, there will be a significant number of hits. And depending on how you do these analyses, you can say that basically 70% of the viriomes are always unknown, which means that they don't hit anything really that anybody's ever seen before. And then most of the microbiomes are actually known. The other important point of that is that 10% in the microbiome are actually just the viruses that are associated with the microbiome. So essentially all diversity, unknown diversity, is in the viruses. The other thing is that all of the interesting things that are changing are in the viruses too. So along here what you have is, this is like family 1, twin 1, that's how that's, so F1, T1. And what you can see is these are the microbiomes here, and then these are different classes of genes that they encode. Okay? So you really aren't very different from each other when it comes to what genes you have encoded by your bacteria. Okay? However, when you look at the viriomes, you can see that they're very different from each other. So everybody has a different viral community living associated with those bacteria. Okay? But the microbes that they're attacking are very different. So this is all your different metabolic functions are actually that make you different from the person next to you is actually really based on your viruses, not on your bacteria. Okay? Does that make sense to everybody? Okay. The other thing about the human fecal viriome is that actually diversity is not that high. So if we go into most systems, what we'll find is that we get about 10 different viral, at least 10 different viral species per each microbial species we look at. When we go into the humans, it's actually much closer to 1 to 1. So it's almost like every bacterial species has one viral species in it. And this is important, I think, because I think this is telling us that this is mostly temperate behavior. And there's some other things here that suggest that that's true too. I'll come back to the temperate stuff in just a little bit, okay? So the other thing that's interesting is that you keep your viruses over long time periods. Okay? So we know that this goes for about 5 years now, where we have the best data set. And what this means is that you acquire those viruses, okay? You keep them. They don't seem to change much across someone over these times, and you can see this in this part of it. So here's the different twins at different times, and they basically look the same at different time points. Okay? But your difference between different people are still very high. So this is low intrapersonal diversity and high interpersonal diversity. And what we wonder is maybe this is actually something that happens in either right at the beginning when you first get colonized by your microbes and your viruses. So this is an example of where a baby got colonized. If you watch a baby, what will happen is the meconium is very sterile, actually. We just don't see bacteria or viruses in there. And then within the first 48 hours or so, you actually see a lot of viruses and bacteria coming into that system. And what we've been able to show by looking at both viriomes and using PCR in different places is that this initial colonization can be maintained for a long period of time. So maybe the first, I guess we're at 4 years now with one baby. So it's actually been able to, those first phage that they got seem to be going on and keeping going with them. So it could be that this initial colonization is important. All right. So the idea would be is that the fecal phage are actually temperate for the most part, right? So if you look at what we find in these systems, we find usually more proviruses this is just the sequence data, or we can find viruses that are like proviruses, so things that have the ability to do that, to behave temperately. And this is across the board. We keep seeing it in all these different systems. And the only time we see that switch is when someone has a severe disease of some sort. So something where the colon isn't working well. Then we see a lot of lytic behavior and a lot more lytic viruses in that case. So the idea here is that really what you've got is you've got a war of lysogens going on in your gut, right? So these are lysogens, they've got prophage, and then they're using those prophage to exclude other strains. And this is some evidence of this. This is a whole series of studies now where we're making mice with humanized microbiomes. Then what we'll do is we'll look at like the transcriptome, for example, and then just say, okay, what viruses are active in the gut, and using BLASTAN and just graph, for example. And what you'll see here is that this would be a prophage genome going along here. And you can see sometimes in the cecum and in the fecal, you've got genes that are expressed from the prophage. And in other cases, what you see is that the prophage will be actually activated a couple to, like 1 out of 3 in the cecum, but all the time in the fecal. And in fact, this seems to be the most abundant case that we're seeing. So in the cecum, you don't have a lot of prophage activity other than a couple genes. Then when you get to the fecal matter, you see the viruses become active, almost like they're bailing out, right? So as the stool is going to get flushed into the environment, the microbes are like, I'm out of here, or the viruses are like, I'm jumping ship, I think is basically that's our model so far. All right, so this is what we know about the human microbiome right at this, and what we're doing, sorry for the gut part of it, and where we're going right at the moment is we're doing these humanized microbiomes in mice, and then we're capturing viruses on them and see if we can get exclusion of different strains by the different viruses going in. The other big question is, does this actually matter at all? And so to look at this, we're following up on those twin studies. So now we've got about 130 twins that are either both are thin, both are obese, or the discordant for obesity and not being obese. And we're looking at to see what we see in those viriomes. So that's the next part of this stuff. And trying to see if there's cool genes that pop up in that case. Is this clear? All right. So I'm going to go on now to our stuff on cystic fibrosis. So if you're not used to, you probably all know this, cystic fibrosis is really caused by this mutation that in the human, where what happens is that you don't get the right, you don't hydrate the epithelial cells of different, like your lungs and different organs in the body. And what happens is the mucus then impacts down on the cells, and at that point it can get colonized by microbes, and eventually you end up with these really horrible microbial communities. And we've been trying to figure out the microbial dynamics of this system and the viral dynamics of this system. And the first stuff, just to give you a background, is just using with sputum. And actually, Christelle and I were just talking about this. This is much harder than it is with other systems. So what we'll do is you homogenize the sputum. You do cesium chlorides to get the viruses, which will be there. That's all the stuff crap. Then we go in and we get the viruses, and then we'll do sequencing. Now, we've got this worked out now, so you can take sputum and you can get the transcriptomes, the DNA metagenomes, and the viriomes, and yeah, all of those fractions. So if you want those protocols, we've actually got them all written up, working nowadays. What we have not been able to do very well is get RNA viruses out of this, these samples, and we're not sure about that, why that is.